but I'm going to speak now really about EMBL and EMBL's history, EMBL's current aspect, and above all, EMBL's relationship to Norway. I am 1/27th legally Norwegian, so I have my discovering my inner Norwegian at the moment. And you can think of EMBL as there being 6 little Norwegian flags planted in Cambridge, UK, in Heidelberg, Germany, in Hamburg, Grenoble, Barcelona, and Rome. And you're very welcome in all of those locations. So this is just a little bit of history of both molecular biology and EMBL's role in this. EMBL is a sort of child of the 60s. That was the time where 2 things were going on in science. So the first was the development of these powerful new physics machines, these machines which developed new particles and new systems, and most obviously led to the development of CERN. And there was an understanding, in particular in Europe, that one could only do that by collaboration between countries. Just the scale was going to be unfeasible. And the second thing then was the kind of development of both structural and then molecular biology at the time, and an understanding that these new types of structured light, very, very synchronous and bright X-rays, very powerful protons and neutrons would give us new insights into the way life works. And that has certainly been true, in particular for X-rays. And the second thing is that there was a new science at the time, molecular biology, and there was tremendous concern that that science was going to be completely dominated by America. And so they wanted to make sure that there was a flagship European laboratory to do that. And because of that, EMBL was founded in 1974 in Heidelberg, Germany. It was founded next door to a neutron source. Now, neutrons are lovely, especially if you're studying magnets. They're kind of useful for life, but they're not tremendously useful. And in fact, the neutron source closed down in Heidelberg. So left up on the hill is this massive molecular biology laboratory there in Heidelberg. But right at its foundation, it was joined by sites. There's a question about whether you brought the biology to the instrument or the instrument to the biology. And for these very big physics instruments, it was very clear that you had to bring the biology to the instrument. And so it was started with 2 outstations, or now sites, at Grenoble and Hamburg, which were the sites of still the two best synchrotrons in the world. Grenoble is the best synchrotron in the world and produces the most synchronous and powerful X-ray light for people to use. And if you go to Grenoble and do your crystal structures, you'll be using one of the beam lines run by EMBL, or if you go to Hamburg. And then we go through the 1980s and really the discovery of all the different things you can create via molecular biology, and understanding life now by thinking about its DNA and mutations of the DNA and how it comes about. And most impressively was Christine Nusslen-Volhard and Eric Wirtschas were at EMBL when they did the Drosophila screens, which won them the Nobel Prize later on. That's not the only Nobel Prize that is associated with EMBL. Indeed, at the same time, Jacques Dubuchet developed cryo-freezing, so the ability to flash freeze biological samples and to do it so quickly that the water does not crystallize. So the water is in this glass state. The water molecules are trapped in that instant, and therefore you do not disrupt the biological nature of the material you have. And that flash freezing was absolutely critical for the development of cryo-EM, which has become such a mainstay of molecular biology, structural biology. Underneath all of this was a theme of DNA and informatics. And this grew and grew over the 80s and the 90s. And at the end of the 80s, the scientists in Europe, particularly in Heidelberg, Ryan was there and some other people were knocking around, were taking journal books and typing in the DNA sequences that were being published at the time. So they're typing them in directly. A human being was reading a paper, typing it in, and then that would go into a computer. This was before the internet. And so what would happen would be that if you wanted that DNA sequence, you would order it, and you could order it in two forms at the start. One was printed books. You could get a complete printout. And so I have seen the shelves of this. It's kind of mad, but brilliant at the time, a real sort of 1980s solution to the problem. Or you could order tape drives. And just when I was just a kid, I was knocking around in this era, and it was very exciting about CD-ROMs. We could have CD-ROMs, and we could send CD-ROMs in the post. So all of this business of sharing data with our colleagues in America and in Japan, it predates the internet. It's quite important to stress that biology's attitude towards data sharing predates the way society has learned to use or exploit data sharing and communications at scale. Now, back in the 1980s, this got out of hand, and the people in EMBL put their hand up and said, we need more space for these people typing this in. It's all getting a bit more serious. And EMBL Council at the time said, well, there's no reason why that should happen in Heidelberg. And so countries were invited to bid for the next site of EMBL. And 3 bids went in. One went in by Germany to expand the site in Heidelberg. One went in from Sweden, which was going to be based in Uppsala, which would have been rather lovely. And the third one was in the UK to place Europe's bioinformatics center next door to Europe's largest sequencing center, the Sanger Center. And that was, the campus was funded by the Wellcome Trust, so the building was a gift from the Wellcome Trust. So the UK did win that, and that's why EBI was present there. And just to finish off the whole story of how the sites came about, at that point it became clearer that it was feasible to run a multi-site molecular biology institution, and we started a site in Rome around mouse biology. It's now focused on neurobiology and epigenetics. And then our latest site is in Barcelona on the beach, absolutely beautiful building, focusing on mesoscale imaging. And you can see now, as we go through to the 2000s, 2015, 2017, and onwards, we have continued at EMBL to be part of innovation. Sometimes that innovation is innovation that happened 20 years ago that we've been recognized for now, and sometimes it has been this incredibly rapid and fast innovation. For example, the creation of the AlphaFold database across all known proteins, applying this AI method to produce accurate structural prediction models for every single protein that we know about. And we believe with our colleagues in DeepMind that we'll be able to stay up to date for that for the foreseeable future. So that's a bit of a history. We have underpinning that history are these 5 different missions. One is excellent research, to perform excellent research and to be that flagship in Europe for molecular biology. The second is to provide scientific services. Now, these are most obviously the synchrotrons, the beamlines for X-ray crystallography. If you do X-ray crystallography, I'm sure you know about these beamlines. They include now the EBI data services, and that has become a linchpin to molecular biology across the world, including Europe, about these data resources that we show to the world. And finally, the newest addition, which I'll touch on later on this talk, is the Imaging Center in Heidelberg. And that is access to super-resolution microscopes and to EM technology for, again, all member states. You can put your hand up and go in a queue to access those machines. And then we have 3 supporting missions. One is advanced training. That means students and postdocs. We have a very vibrant student and postdoc rule. And back in 1974, the countries that founded us said that you must not keep people. And so we are not allowed to hire from our student population any postdocs. and we're not allowed to hire from our postdoc population any faculty members. And our faculty members must leave after 9 years. There's a small set of people who are considered tremendously important or lucky who go beyond those 9 years. But legally, over 90% of people must leave beyond 9 years. And this means that EMBL is always turning over and always producing people. It also means for whatever career stage you're in here in Norway, there is a career path that involves EMBL. So if you're interested, there will be a studentship or a postdoc or faculty position. And I'm really, really keen for you guys to think about your career path as weaving into EMBL and then coming back, probably to Norway because it's lovely, but coming back somewhere else for this. Innovation and translation, again, I'll touch on it, which is about how we interact with companies. And then finally, integrating life sciences, and that's particularly true about our role with Elixir in data. And this is a picture of our lovely member states. We're a bit like Eurovision in the sense that Australia wants to be European here, so Australia is down at the bottom. It's the vast majority of Europe. Slovenia is a bit of a holdout, and we will get there at some point, I hope, joining together for this. And this slide is a busy slide because it also indicates all the partnerships we have. And these are partnerships which are local to the sites that we have, or partnerships which are remote and have set up under the EMBL model. And those include the Norwegian Centre for Molecular Medicine, NCMM, in Oslo, which has been a tremendous success there. And that's part of the Danish-Nordic partnership, but also includes places like Brain City in Poland. And this, again, is just to remind you of these 6 different sites. So our headquarters are in Heidelberg, Germany. It's also our largest experimental lab. The second biggest site is EMBL-EBI, just south of Cambridge, on the same site as the Sanger Institute, and we're completely dry there. And then we have Hamburg and Grenoble, which are the 2 synchrotrons, Rome, which does effectively mouse biology, epigenetics, and neurobiology, and Barcelona, which is about tissue biology. So we have many, many different links with Norway in many different ways. This is a 3 that we're particularly proud of. So a publication and analysis of microbial sequencing done jointly with NTNU and colleagues in EMBL. This rather wonderful business of, and I think this is with the SARS Center, this business of looking at the diversity of metazoan life in marine animals. And this is looking at sponges and the different types of cells in sponges. And these rather small marine organisms are kind of brilliant because you can put the whole organism into an EM device and look at them there. And on the right-hand side, the development of the Federated European Genome Phenome Archive. So this is about the responsible sharing of clinical data between collaborations. And Norway has been one of the founding nodes of this federation for delivering this. And as you saw on the map, we have 2 different partnerships here. One is very much on the medical side, the NCMM in the University of Oslo, and the second one is the SARS International Center for Molecular Marine Biology to do this. And again, the partnerships differ, but one of the things that the partners, I know many partners, appreciate is the ability to have and model their career paths across EMBL of this turnover. Not every academic institution can have a turnover model, but some academic institutions having a turnover model ensures that we have throughput of people and new ideas entering the field. Our new leader, Edith Hurd, my boss, came to EMBL from Institut Curie in Paris, and she challenged us to do even more, which was a bit daunting. I have to admit, the first time I heard what Edith wanted to do, I was like, are you sure? But it has been really invigorating, and that is to keep one foot wedded to this atomic, molecular, cellular world, but to work in collaboration with all these people outside of the organism, so the organism in context. So this is to think about how organisms interact with other organisms and with the environment. And I have a particular passion for one organism. Those are humans, and they're quite unique in the way they interact and create environments, but all organisms count here. And that, by the generous awarding of money from EMBL Council, means that we can realize that. It is not a small ambition, molecules to ecosystems. Just be reassured, we do not expect to do all of this within the walls of EMBL, by definition. Rather, we want to be, again, a flagship and a place of collaboration across Europe to deliver this. And there's lots and lots of interesting environments in Norway, both for humans and for all these other wonderful things, which I'm sure I'll meet up in Tromsø. So this is this new program. It is bold. It meets a lot of societal challenges, both health challenges, but also global planetary health challenges, about how we think about the planet. And again, collaboration and interdisciplinarity has always been a big part of EMBL. We've always had a lot of physics and maths alongside the molecular biology that we do, and that will only get deeper and deeper over the coming years. So these are some of the things that we expect to impact over time. Like many scientists, I celebrate the times we create impact in the world and we do something that changes things. I get a huge buzz out of making people healthier. I think it's amazing. But that's not actually how I organize my science. I organize my science by curiosity and what drives it. And these are the things which we believe we're going to make an impact on, but it's not a given. And I suspect our biggest impact is something that we cannot write down now. And that's the way this goes. So looking at ecosystems, but in particular the carbon cycles and weird and wonderful new carbon fixation routes, which is present in life, and that's very clear in the microbial systems that we've been looking at, is there's quite a lot of diversity in the fundamental elemental cycles of life. Thinking about biodiversity and thinking about how we measure it, how we capture it, and then how we use it in our understanding of life in evolution. Thinking about the impact on humans and disease, in particular the interaction of microbes, and thus we create an environment for a large number of microbes in our skin and our gut, so we are an environment as well as being people. And then one particular headache that we've got is like a slow burn pandemic, is antimicrobial resistance. I don't know if anybody knows this, it's very depressing. We are running out of effective antibiotics, and if we don't fundamentally change our ability to find new antibiotics or manage the way bacteria are getting around our current antibiotics, we could go back to the 1910s, 1920s, where we did not have effective antimicrobials. And that would be a very bad world to go back to. So that's quite an important thing for society to get right, because it's a darker place without that. And just like here, we are going to do transdisciplinary research. Now, so we set this up with these core of the cellular biology, structural biology, developmental biology, and computational biology that we've always had, but we've added some other areas here to stimulate these things. I'm going to step you through the newer areas, but I just want to emphasize that the central part is still just as big and vibrant as ever for this. And just to mentioned that this 9-year turnover of groups means that the actual individual research that's happening at EMBL is always changing and refreshing. So if you knew EMBL in the 1980s, at some level it's still the same physical building and there's aspects of the same culture there, but there are very, very few people who were around then, now, or in the 1990s or the 2000s. So stepping through this, we have the planetary biology transversal theme to look at the planet. This has in some sense been piloted 5 years ago by this wonderful ship, research ship called Tara, manned by gloriously mad Frenchmen, sailing the oceans and sucking seawater through filters and capturing microbes and viruses and plankton on their filters. There are way more viruses than anybody ever thought there were going to be in the ocean. Like, way more is one sort of surprising take-home from that. But the bacterial diversity is mind-blowing in the ocean. They want to do that again, but they want to do this around the coastline of Europe, and they want to do this with a EMBL truck, the Trek mission, going alongside the coast, beside the boat, so that we will be sampling the ocean or the beach and the bit of sea close to the ocean, close to the beach and the environment all at the same time. So they've done this. They've done a pilot in Naples. It's definitely doable. There's quite a lot of interdisciplinary work here between ecologists and molecular biologists. Interestingly enough, they think the genomics is very routine and boring. I find the genomics quite exciting. But they're going to take cryo-freezing equipment on the truck, so they aim to freeze a lot of these animals as well in a format that they can put into the electron microscopes later on. So freeze these weird and wonderful living organisms as well. And there's many other projects here, including our collaborations with the Earth Biogenome Project, which links up to sequencing all the animals on the planet. Again, one of those rather ambitious statements that you think to yourself, is that doable? So one, not every single individual, obviously, but one of every single species on the planet, every single animal, is a feasible thing to do. And a number of projects have started to do that in a number of locations. I would encourage, I would say Norway, but it's really the Nordics, to think about that as something to do across the Scandinavian peninsula. As I said, one of my favorite animals is humans. They're great animals, bipedal organisms. Humans are kind of interesting for a number of reasons. So one of them is they're so important for medicine. So understanding molecular biology of humans and understanding the physiology of humans is an absolute sine qua non for medicine. They also have this incredible ability to walk into phenotyping centers, which are called hospitals, and you can phenotype these organisms almost for free if you're a researcher by tickling the hospital just right there. It is a remarkable property of humans, and it means that some fundamental pieces of research we do basically around humans. My favorite humans are wild Danes in their Danish environment, or wild Brits in their British environment, because those are the two best tracked sets of humans around. But humans interact with an environment, and there's two types of environment for humans. So one is what you normally think about the environment, which is sort of temperature and chemicals and things like that, and that's quite an important environment and has an impact on everyone's life. The other environment is this social environment, the environment we set up between us. And it ends up being that every time you do research on humans, it immediately becomes obvious that this social environment is a huge part of how humans develop and how they, for example, access healthcare and what they do and all these other things. So if you want to understand human health, you have to understand the social environment. And that's interesting. It gets you into psychology and psychiatry and those medical domains. And it also gets you into neurobiology. Why and how do humans think the way they think in the setting up of this? And then the other interesting thing, as I mentioned, is that humans themselves are an environment. We're an ecosystem, and we have a very active ecosystem of gut bacteria. There is now 1 or 2 medical procedures where replacing the gut microbiome is an effective treatment for particular things. So there are 1 or 2 cases where this really works. There's an awful lot of snake oil and pseudoscience also in this field. And so kind of impacting this between, understanding what is going on, understanding our gut microbiome, understanding what is science versus pseudoscience is tremendously important. And that is closely related to this other transversal theme, infectious biology. Edith always stresses, we were going to do this before COVID. Unsurprisingly, when COVID happened, so much of our effort went around responding to the virus, and we did a lot of data sharing enablement for COVID, which I'll touch on, I hope, later on. But as well as the microbiome and the way they interact in the gut, there's also this business of really understanding how parasites, viruses, and bacteria contribute to diseases. And this is the work by Maria Birney here in the second thing of a tissue-engineered vasculature. So she can make artificial blood vessels in the lab. And she can make artificial blood vessels that look like tissue and artificial blood vessels that look like brain. And therefore, she can study how parasites interact with these blood vessels. And she can do genetic manipulation of the cells before she makes those blood vessels. And so there's a completely new toolkit coming up, which is the ability to create sort of tissue-level systems that replicate parts of the human system for infection biology. We love microbes so much that we have a particular focus on them. And so this is the business to look at how bacteria interact with their environment in all sorts of different ways. And one important thing to realize is there's an awful lot of microbes that really don't like oxygen. And so when you do something in the lab, I mean, people forget we live with oxygen all around us, and it's totally dependent for life. There's a chunk of really important life that finds oxygen highly, highly poisonous, and they just cannot be exposed to it whilst they're functioning. And so that is why you need quite a lot of focus on being able to, if you want to study those microbial ecosystems, which might be in the soil, or they might be in our guts, or they might be in cows' guts, you have to deal with this anaerobic, very strictly anaerobic situation to be able to study this. And that requires quite a lot of different mindset and technology around it. And then there are 2 more interdisciplinary areas here. So one is bringing in more mathematical thinking. Now, EMBL's always had quite a lot of maths and computer science in it, but this is formalizing that process even more and having a sabbatical program for mathematicians. I have no idea whether there are mathematicians. Are there any mathematicians in the room? Perfect. If you're a biological mathematician, this is for you. We hope to run a sabbatical program where we can bring in mathematicians for a period of time to be alongside and see the problems that biologists have. And there's a really interesting interplay here between AI and mathematical theory, where AI, which is linked both to this theory world and to the next world, data science, ends up being a great way to understand molecular biology, but often you need to have the right mathematical framework to present the problem to the AI machine. And so the mathematics of this is quite important. How do I represent an organism's 3-dimensional layout in a sensible way without going bonkers, basically? You need a good maths framework for that. And that links to this other side, data sciences, again, EMBL has been doing this a very long time, in particular EMBL-EBI, which is really the anchor institute, not just for Europe, but often for the world, for these data resources. So the reference human genome, the reference mouse genome, these protein structures and all these other things. But we want to extend that across EMBL and make it part of EMBL in all the different ways, and we want to share the expertise about how to do this with all our member states and our partner institutes. Now, I know this is a big part of what you do here in Elixir Norway and other things. So definitely, this is all part of that same business of bringing more computational biology, quantitative biology, and data science into biology in a productive way. And one of the huge success stories has been this collaboration with DeepMind on AlphaFold. We started working with DeepMind about 5 years ago when this was an idea, and when they were looking around for interesting problems to solve. I must stress that the actual algorithm itself is really John Jumper and Demis Hassabis in DeepMind. So they're the people who came up with this. But they themselves will acknowledge absolutely two things. The first thing is that this problem was unsolvable unless there was the deep and diverse protein structure database present in the PDB, so the protein data bank. So if we had not been collecting protein structures from the 1970s onwards, they would not have been able to solve this problem. And the second rather wonderful thing is that they collaborated with us to provide everything out there for free, which was rather generous of them, and it's actually non-trivial to do. But let me just tell you about this problem. Many of you will know that the protein sequence very often determines the unique three-dimensional structure of a protein. So if you just have the protein sequence, you stretch it out, you put it in the right solvent, you give it the right ions, very often the protein sequence will refold to being a particular protein structure. Now, that's not true for all protein sequences, and it's not true for 100% of a protein sequence. There's some floppy bits and some disordered bits, but there are bits in the middle which we call globular, which do this. So DeepMind solved this problem. Now, they kind of, at one level, cheated in the sense that they used evolution. So they don't take just one protein sequence. They take, usually it takes at least 10 or 20, but these days it's very, very easy to get an evolutionary spread of 10 examples, just 10 different mammals, 10 different vertebrates. It is not a hard thing to do. And it's not really cheating in the sense that everybody had decided that this was an okay thing to do for a very, very long time in this game. And then they developed an AI program. It is the most complicated AI program that DeepMind, who arguably founded AI, have ever developed. And it's really interesting about the details about this. They had to reimagine the way they did certain things to solve the problem. So there's actually quite a long and complicated story about how they developed the AI system. But eventually the system produced confident predictions. Now, why are people so confident in this? The reason why is that John Molt in 2000 set up a competition for this problem. So every 2 years, this CASP competition happens, where John goes around and he asks his colleagues in the structural biology community, which proteins are you solving the structure of now? Oh, sorry. Apologies. And I'm out of time. I'm going to have to speed up, aren't I? Anyway, they are confident because of that competition, and they can solve these structures. I think I'm almost done. And we are doing AI in different areas. So one is very much image analysis. One is genomics. This is my lovely Danes, and doing AI on the Danish healthcare system to crunch the numbers there about risk prediction for this. I talked about those things. I just want to finish up touching on a couple of things. So one is training. We've always done this business of doing pre-doc courses, but we've also done, so that's about PhD students. Please recommend to your undergraduates the EMBL PhD students from a very broad range. You can be a physicist, a mathematician, probably not a historian, a molecular biologist, wherever, if they're interested, and if they're interested and they're enthusiastic and they want to learn, they really, really have a place to learn at EMBL. But we've supplemented this, and in particular, there's a new program for technology developers and engineers. So this is very much not the PhD track. This is an engineering track, and was stimulated somewhat by the scientific engineering in physics, and this is to allow people to be trained sort of on the job in software engineering or technical engineering to deliver these scientific services. And then touching on innovation and translation, we do spin-off companies. So, for example, one of them is this one from EMBL Rome. We actually have a great line in X-ray image detectors through a spin-off company in Grenoble. But we also do very deep public-private partnerships, and one around databases has been Open Targets. This has almost raised £100 million, 100 million euros, probably has raised 100 million euros over its lifetime. And these are pharmaceutical companies putting their hand in their pocket and bringing money to the table to accelerate parts of the research that they want to see happen in the data sciences. And if you do drug discovery, this is an incredibly good informatics platform for doing this. And then we've got these technology development projects. For example, some of the newer microscopes from Zeiss and Laser come from collaborations or spin-outs licensing from EMBL. And then we do this integration of the life sciences. We work with lots and lots of S3s, but I'd particularly highlight the Elixir aspect, which Linné also highlighted on bioinformatics. The hub of Elixir is hosted by EMBL in the same buildings as EMBL-EBI, and is developed there. And I can see that I have really badly overstepped. So this is my last and final slide. Please, please, please engage with our program. Please recommend the PhD program to students or colleagues. If you're a postdoc, please, and you're interested, you're a PhD student, you're ending, or you're a postdoc, thinking about your postdoc, please come and apply for being a postdoc. You can design your own postdoc via the iPod program, which is a bit daunting, but we can help how you design that. And then in particular, use our services. So come and exploit us in Heidelberg. So thank you very much.